Favourite colour? What's my favourite colour, Miriam?
Orange.
Orange.
And yellow.
And yellow. Orange and yellow are my favourite colours. And what are your favourite colours?
Pink and purple.
I like clear colours, like clear yellows.
Well, I like oranges. Blue and purple. Blue. Red. Because my favourite team playing red.
Our whole world of colours, around 5 million hues, is created from combinations of just 3 primary elements: red, green and blue. We're trichromats.
That's the nearest to the lilac. It's a little bit lighter, but it's as close as we're going to get, I think. But the yellow is in the other. I'm so particular. If I'm matching something, it's got to match. To me, it's glaringly obvious that 2 greens are not the same greens. But everybody else says, well, they're perfect.
But Lynn lives in a colour world that's different from yours or mine. She sees enhanced colours, influenced by subtle changes in hues.
As I said over there, it's the nearest you're going to get in this colour, but it's lighter. It's definitely lighter, but it's the nearest one we can get.
It's hard for those of us who live in a 3-dimensional color world to imagine what that would be like, but I think it's fair to say that colors that you and I would not distinguish look different to that person.
I see differences in the same shade, or the same colour, I suppose. I see different shades of the same colour, or probably more of them than you. But it's difficult for me to explain because I've always seen this way.
Our colour world, our own experience of colour, is unique to each of us. Colour vision is subjective, so we can never know exactly how a colour looks to someone else.
I think turquoise is more of a bluey colour. And more of a greeny colour. Bluey colour. It's sort of a greeny colour with a hint of blue.
I guess some people say it was green, but yeah, blue, sea.
Probably nearer green than blue.
So, for instance, if you go to the paint store and you look at the little tabs of paint, one person might say, these two look nearly identical to me, whereas another person will say they look different. And that's very certainly a clue that the internal representation of the world is different.
So we can never know exactly what Lynn sees.
People say to me, well, what does it feel like? You know, what do you see? I mean, how do you explain to somebody who's never had curry in their life what curry tastes like?
Colours begin simply enough as physical entities, wavelengths of light of different energies. Colour vision is our experience of the effect of wavelengths of light on our retinas and the way their impact is interpreted by our brains. The 17th century physicist Sir Isaac Newton first proved that this is the true nature of light when he was 23. He improved his understanding of optics throughout his life. Newton split white light into an infinite number of rays, an infinite number of different coloured hues.
Rays, to speak properly, are not coloured. In them there is nothing else than a certain power to stir up a sensation of this or that colour.
He portioned these hues out into a 7-colour rainbow. He was right. He was also wrong. He was publicly criticised by his lifelong rival, the physicist Robert Hooke, and by the popular science writer Gautier Dagoty, who published one of the first full-colour prints.
And in one of these, he actually prints a Newtonian spectrum with just 3 colours inks and produces a very good gradation of many hues. And his argument was clear. Since he can produce all these colours just by mixing 3, Newton must have been wrong. There must just be 3 primary kinds of light.
It was another 200 years before Thomas Young, a third physicist, settled the argument. He proposed that there are 3 kinds of resonator in the eye. His resonators are our 3 cones. Each one of these 3 cones is sensitive to either red or long-wave light, green or medium-wave light, blue or short-wave light.
Right, we seem to have a nice preparation here.
Each kind of cone can be identified by shining light at known wavelengths onto it. Professor Jim Bowmaker works with cones from fish retinae, but the principle's the same for us. The cone shows a peak in activity at the point, the wavelength, at which it's sensitive.
We can draw out the absorbance spectra of the 3 cone spectra for the human cones. Here we have the Al cone, or the red cone, with a peak at about 563 nanometers, the M cone, or the green cone, with a peak at about 535, and then the much shorter S cone, the blue cone, with a peak at about 420 nanometers.
The same tests give some clues to how Lin is different. We have 3 kinds of cones with pigment sensitive to one of 3 wavelengths. Lin has 4. Lin's extra pigment is sensitive in the green part of the spectrum, skewed towards red.
And I can add on a 4th cone class here, an anomalous pigment, say at about 540 nanometers. And these are the females that potentially could be tetrachromatic.
Each photopigment, each light-sensitive protein, is an opsin.
As a single photon, a single particle of light, hits this one protein out of millions, and it leads to this firework of chemical responses in the cell. So this is a very beautiful system. It's built for a huge amplification. It's built to act very quickly, and then it turns off very quickly. So you can think of it, the retina, you can think of as like a slate that's wiped clean after each time it's used. Of course, if it wasn't, you'd be stuck viewing the first thing you ever saw, namely your mother looking at you. But as it is, you can play tennis because you can wipe the slate clean after the ball goes over the net and you're ready to see another scene.
The eye is often compared to a camera, but it's much more complex than that. In the 1950s, Edwin Land invented the Polaroid instant photo-making process. His invention came out of an almost accidental discovery. He found that our eyes can recreate the whole range of colours in black and white slides, using just 3 clues. He took 2 pictures, one through a red filter and one through green. He printed them in black and white. Then he put the filters back. Mixing pure red and green light in the right amounts creates a sensation of yellow, even when there's no yellow wavelength light. This is the result of additively mixing spectral primary colours. All the primaries mixed together equally will make white light. He called his explanation his Retinex theory. It shows the visual system doesn't simply record colour by monitoring wavelengths of light, it actually constructs colour. So Newton was right about the nature of coloured light, but he was wrong about coloured objects. Objects obey different rules from lights. Objects reflect some wavelengths of the light and absorb the others. The reflected wavelengths are the ones seen. That's the reason why Hooke was also right. It is possible to print a full spectrum from just 3 colours. That colour of light or objects is constructed by the visual system gives the next clue to Lin's condition.
Now, each of these kinds of cone is itself colour-blind. It just collects photons, and it doesn't know the wavelength of the photons that it's absorbing. So, although it maximally absorbs in one part of the spectrum, it does absorb photons, if there's enough radiance, in other parts of the spectrum too. So to find out about colour, the visual system has to have a way of comparing the rates at which photons are being absorbed in different parts of the spectrum.
So if you look at a red apple, for instance, the light that is bouncing off that apple tends to be enriched in longer wavelengths and less in shorter wavelengths. That's an intrinsic property of the skin of the apple. Now, what you, as a potential eater of that apple, would like to know is whether it's red or whether it's green. It perhaps tells you about whether it's going to be good to eat. How can you figure that out? How can you figure out which kinds of photons are coming off that apple? Well, if you have a series of pigments in your eye, which we do, which absorb selectively different wavelengths of light, then you simply have to ask which ones of those pigments are active, or which ones are more active than the others. And in the case of that red apple, the pigment that is most sensitive to red light is the one that is most activated by the light coming from that apple, and the one that's most sensitive to green light is somewhat less activated, and the one most sensitive to blue light, less activated still. And your brain gets that ratio of activations and says, aha, this means the apple is red.
So differences in the activities in each kind of cone, red sensitive or green sensitive, are compared. It's more red or it's more green, and this ratio dictates the precise hue we see.
So Lynn, this is a very easy test to do. The computer's going to show you a coloured ring like this on the screen, and there's a gap in it always. And the gap can be here, or here, or here, or here. And I'm going to give you this button box.
Lynn's taking the very latest version of a classic sight test. She's very quick at seeing the outline hidden in greyed hues of the same primary colour. It shows her discrimination is very sensitive. Her responses are plotted on a CIE chart.
What is it on the computer that shows that I see differently to somebody else?
What we've got here is a graph that allows us to show all the colours that can appear on a computer screen. So the purest red a computer can produce is here, the purest green here. And
Lynn easily sees the coloured tint to each grey, so her scores are clustered together in the centre of the chart. Someone with normal colour vision may show a result that's far more spread.
All the hues that we can experience, this wonderful variety of hues that someone with normal colour vision enjoys, are generated by the visual system just comparing the rates at which photons are absorbed in these three kinds of cone. And differences of as little as 1% in these ratios are seen by us as different hues.
This information is passed up the chain of cells. Cone cells on the inner plexiform layer of the retina synapse with midget bipolar cells in the outer plexiform layer. These midget cells in turn connect to ganglion cells and on up to the optic nerve. At each layer, input from the receptive fields of neighbouring cones is compared. At each stage, the input is weighed up, more red or more green, and a decision taken on the ratio of one to the other, in trichromats. The 19th century physicist John Dalton was a dichromat. He was the first person to realise his own colour vision was different from others.
When a young man, he noticed that the flowers of Pelargonium zonale looked to him sky blue by daylight, but pink by candlelight. And he was astonished by this change in apparent colour, and he tried to attract other people's attention in it. Now, his own brother agreed with him, but no one else did. And they soon realised that their colour was unusual. Now, Dalton supposed that to account for the change in the appearance of the pelargonium, there must be a blue filter in his retina. And since he couldn't see anything blue from the outside of his eye, he supposed that it was the vitreous humour of his eye that was bluish.
Dalton was so convinced he had blue eyes, literally, that he left his eyeballs to science. When he died, one was dissected by his assistant Ransom.
He just scraped off the back, the sclera and the choroid, so that he could view objects through it. And he assures us that scarlets look scarlet and greens look green through Dalton's eye.
So it wasn't that his eyeballs had a coloured cast, as Dalton had suspected. The secret lay not in Dalton's eyeballs, but in his genes. Professor Mollen could prove this, as Dalton's eyes still exist.
A year or two ago, David Hunt and I were able to extract DNA from his eye and show, in fact, that he shared the same kind of physiological explanation for his colour blindness as do most dichromats in our population. He was lacking the middle wave receptor of the eye.
The code for the protein structure of each pigment is held in the genes by amino acids. Professor Nathan sequenced the genes and uncovered their codes. He found that the instructions for red and green photopigments are surprisingly close. He's been tipped for a Nobel Prize for this exciting work.
If you look at the human red and green pigments, and these differ quite significantly in their absorbance, there are only 3 amino acids that are important for determining that difference. And clearly evolution has found them and has changed them just so to make those pigments absorb just as they do.
That the genes code for photopigments with different sensitivities is the blessing that gives us colour vision. That these codes are so similar is also the reason why sometimes coding goes wrong.
It's because these genes have remained together since presumably the original duplication event that so much mischief occurs in human colour vision. Because the 2 genes are close together, it's possible for the genes to be misaligned at meiosis and for one chromosome, one X chromosome, to end up with 3 copies of the genes and one to lose one of them, for instance.
It's brown. I know. They said green.
David has lost one copy of the gene, the one coding for the green photopigment. He has just 2 colour channels, long wave or red, and short wave or blue. He's a dichromat. His classmates have copied the colour codes accurately. David makes mistakes. He confuses browns, a red and green mix.
My mummy can speak Which one of those did you think it was? Did you think that I thought that was... Yeah, that's C.
Is that brown?
Yeah, that's brown. Go, David.
Why did you colour that blue?
David is a deuteranope. His retina would look like this. In the alternate form of dichromacy, protanopia, the red pigment cones are missing. Together, these conditions are shared by 1 in 100 males. At 10, David's coming to terms with the idea that his experience of colour is different.
So, Tim, this is a very easy test to do. What I'm going to do is show you on the screen here a coloured ring, and it's got a gap in it, here or here or here or here.
Lynn's son Tim's colour perception is also differently constructed. He's a trichromat. He has 3 colour pigments, but he's an anomalous trichromat. The pigment sensitive to middle wavelength isn't missing like David's, but it is shifted towards red. It registers a hybrid of green and red, and it sends this skewed information up the line for processing. This is due to anomaly. Again, protanomaly is the opposite. Red shifted in sensitivity towards green.
What their perceptual world is like, I do not know. I can't tell you. There is a great variability between these types, so within these groups of either deuteranomaly or protanomaly, people wouldn't necessarily agree on their percepts either. They would have different views of the world. But if you look at a protanomalous or deuteranomalous person colouring in a book, you would certainly do not agree with what they're colouring in, but they would confuse some colours in the spectrum depending where their remaining 2 middle to long wave pigments are.
Males are affected many more times than females because the amino acid codes for pigments are sex-linked. They're held on the X chromosomes which determine our sex. Females have 2 X chromosomes in every single cell of their bodies, so a mistake on one chromosome of the pair can be compensated for by the other. Males have 1 X and 1 Y chromosome, so a mistake on the X chromosome goes uncorrected.
This male offspring here has inherited the normal X chromosome and is therefore colour normal. But this person here has inherited the X chromosome that carries the hybrid gene, which then makes him into a deuteranomalous subject or person. And this is exactly the phenotype that we found in Lynn's son, Tim.
What I've got here...
Tim has a hybrid form of the green and red pigments, so his colour vision is skewed. Professor Mollen can test this rigorously using an anomaloscope. This is a colour matching test. Tim's asked to mix the amounts of red and green wavelength light in the right circle to match the control colour in the left one. The relative amounts of red and green light Tim sees, the ratio of red to green, is shown up by the match he makes. Because his green-sensitive pigment is a hybrid, his mix is off. And it was the exact nature of his mistake that led Dr. Jordan to Lynn.
Yeah, that's okay. Ish.
How's that?
It's about finding sons who have very good discrimination left, given their anomalous trichromacy, which then would be good evidence for us that their mothers would have also pigments which are separated enough from the spectrum in order to be used by the brain. And it is a little bit like looking for the needle in the haystack.
Anomalous trichromacy is the most common form of colour deficiency. It's found in about 6% of men. Men express the condition. Their mothers are carriers. Lynn's passed the hybrid form to Tim, so she carries it. But she has very good colour discrimination, so she must have all the normal photopigments and a hybrid form as well.
She has the normal S, normal M and L, because the pigment genes lie on the one chromosome carrying these normal pigment genes. And she also has expressed in her retina the intermediate pigment which makes her son into a deuteranomalous person, so she ought to have 4 types of colour pigment.
So Tim's condition shows Lynn meets the first criteria for tetrachromacy. Overall, Lynn will have 4 pigments in her retina: long, medium, short and shifted medium wave. But that's not enough on its own. She needs to have sufficient hybrid cones for this wavelength to register in the mix of red and green wavelength cones. Again, this is dictated by her genes. In every cell, including cones, Lynn has 2 sets of chromosomes, so she'll have codes for both normal and hybrid forms of the pigments. But only one code is expressed in any one cell or cone. The other is switched off.
And that seems to be something of a coin toss between the 2 X chromosomes. Any given cell will express one, but its neighbor may express the other one. And in the retina, we know that's the case, as it is elsewhere in the body. And so a woman who has 2 different kinds of pigments encoded on the 2 X chromosomes is actually going to have in her retina a mixture, a mosaic of cells expressing one or the other.
Okay, Lynn.
So the acid test is, does Lynn have enough hybrid cones switched on and active in the system to make a difference?
Closely as you can to the field on your left, which is a standard yellow.
That's actually the closest I can get, but it's not...
The really interesting question is if she has 4 types of cone or 4 types of cone pigment on the level of her retina, what does that do to her visual system? Can she discriminate more colours? Does she have some advantage? Or is her vision just like that of a normal person? Right.
But if I go...
Her results show the ratio of hybrid to normal pigment cones is both high enough and their wavelength sensitivity distinct enough for their information not to be lost.
I think it's quite plausible that our brains are complex enough to handle that additional information. So the only question really is, at the front end of the system, can the retina add another cone class, and can that wire up correctly and feed that information to our brain? And given the modularity of the system, I think that's also plausible.
There's yet another criteria that Lynn must meet, a 4th circumstance that makes her a tetrachromat. Not only does she need enough hybrid cones, but there need to be enough of these in the part of her retina where cones have their own channels. Professor Bowmaker agrees the system is flexible enough in principle.
And we can see this could happen in the central fovea because these cones which contain the anomalous pigment will have their own midget bipolar cells, their own midget ganglion cells, so they still have their own private spectral line. However, we don't know whether that line is wired in to a traditional AL or M channel or forms its own new channel. And so if they do have their separate lines and they're maintained, then we may have tetrachromacy. But on the other hand, if they're wired in either to the original AL or M, then although the sensitivity of that channel may change slightly, we will still be reduced to trichromacy. And I think this is still a very open question.
But if I go...
Lynn has the equivalent of perfect pitch. She can hit the same colour note, the same hue, each time.
Now, if you consider a tetrachromat who has an extra pigment in this region of the spectrum locus of this type of color space would not be a straight line, but would be somehow curved.
And that, in effect, pinpoints a place outside the area of the CIE chart, in a fourth dimension of colour space. So the criteria for tetrachromacy are many and stringent. A true functional tetrachromat must meet all of these criteria. It's possible that Lin is one of very few.
We had 7 carriers of protonomaly and 7 carriers of deuteronomaly. Most of them behaved in much the same way as the normal, except for 2 carriers of deuteronomaly who really made very specific, very well-defined matches completely outside the range of the normal matches in a different space.
And tetrachromacy is possible because of the very way our visual system is built.
It's a remarkable fact that those different pigments that are used for color vision arose by evolutionary duplication from a common ancestral pigment. That is, the proto-organism, whatever it was hundreds of millions of years ago, had one light-sensing pigment. It used this same chemical mechanism of a little flip-flop of the chromophore. But the gene that encoded that pigment then duplicated, and it gave rise to copies that differed somewhat in their sequences, and part of those differences resulted in a difference in the absorbance of the encoded pigment. And then the organism, whatever that organism was many, many years ago, had the possibility of comparing the degrees to which those different pigments were stimulated, and that was the birth of color vision.
2 kinds of evidence suggests our visual system is designed to take out and add in extra channels as it suits, like building with Lego blocks.
The early mammals were nocturnal, and they only had a very simple colour vision. They were dichromats. They had one main type of cone with its peak sensitivity in the middle of the spectrum, which they used for the really important things like detecting flicker and movement and form. And then in this array of receptors in the retina, there were a few sparse shortwave cones, and by comparing the amount of light that was absorbed in these shortwave cones with the rate at which photons were being absorbed in the middle wave cones, they got a basic color vision because they were basically comparing the balance of wavelengths between the shortwave and longwave part of the spectrum. And this ancient system of color vision survives as a separate pathway in our own visual system. It's depicted here. So this ancient pathway...
Then one channel, the long wave channel, splits again to create a third.
Now, about 30 million years ago, when our ancestors were arboreal, living in trees, and particularly trying to find orange or yellow fruit amongst the dappled foliage, a second subsystem of colour vision evolved. We don't know exactly when. What happened was that there was a duplication of the gene that's on the X chromosome that encoded this middle wave photopigment. In the course of evolution, the peak sensitivities of the long and the middle wave cones separated, and there's a second subsystem of colour vision that we now enjoy, which compares the rates at which photons are absorbed in the long and the middle wave cones. It allows us to discriminate between reds and oranges and yellows and greens.
Evolution of a 3-channel system gave our nocturnal ancestors an advantage as they moved to daylight living. Clearly, this flexibility has its uses, and flexibility gives variety that is present and useful today.
It's an intriguing observation that individuals with dichromacy are actually better at certain visual tasks than trichromats are. For example, they are less confused by camouflage, and this has been known in the armed forces for many, many years. That is, if you want to break camouflage, a good way to do it is to have a dichromat look through the binoculars and look for the object of interest. There's every reason to believe that in a natural situation, say in a group of hunters, perhaps in Africa a million years ago, that if one of them had dichromatic rather than trichromatic vision, that individual might be at an advantage in finding certain kinds of prey. And so we imagine that there might in fact be a positive selection in some circumstances for individuals with variant color vision.
So the intriguing question is, if 2 channels gave rise to 3, in Lynn, are we seeing the channels hybridizing again to give 4? Again, there's an evolutionary precedent. Our nearest ancestors are Old World primates, the apes. They're trichromats like us.
The trichromacy in primates, what it provides primates with, so obviously including ourselves, is the ability to discriminate in the red-green region of the spectrum. Now, whereas Old World primates have this gene duplication, so they have separate red and green opsin genes on the X chromosome. New World primates, with one exception, have a trichromacy based on a single gene, but a single polymorphic gene.
One gene which comes in 2 forms, each specifying a pigment sensitive to a different wavelength. Between them, these forms create 2 pigments which act like our red and green channels.
The curious thing is that in most New World monkey species, all the males are dichromats. A subset of the females are also dichromats. That will be the case if they happen to inherit from their parents 2 identical genes. But of course, equally, a subset of the females will be trichromats, those that inherit 2 different forms of the gene from their parents. And the mere fact that, of course, we find trichromacy in the population, and this polymorphism maintained, therefore, in the population, that can only happen if it has a selective advantage. So we have essentially a heterozygous advantage, a selective advantage, but of course operating only in the females.
Some females have an extra hybrid form, just like Lynn. One chromosome expresses the first form, the second chromosome expresses the second form. But in evolution, the acid test is, does it give an advantage?
We think that these candidate tetrachromats do have an advantage if they have to discriminate colours, for example. And Lynn's behaviour on this specifically designed test has shown that her colour vision actually differs. Now, I think that the difference is much more subtle if you compare it with a big step from going from dichromacy to trichromacy. But subtleties, nevertheless, can give an advantage. If such a putative tetrachromat has children who show slight changes in skin colour, for example, showing pink flashes or going slightly pale or slightly show a slight bluish tint, she may be able to pick up these subtleties much quicker than a normal person.
Among our ancestors, orangutans are known for having a very good memory for fruit. They hold a map of all the fruiting trees in their territory, and they have good colour vision like us. If the 2 are related, it's possible good colour memory could also offer an advantage.
If there's an extra signal in the retina, and this signal is being kept segregated up to the level of the cortex, we may also assume, although we haven't tested it yet, that, for example, this signal will be processed by colour memory differently. And so these women may display a different colour memory, say it might be better. But we have not tested it yet. But it is one of the obvious things to do.
Right, the little pots of yellow are down here somewhere. I know I've seen them down here somewhere. Oh, here they are. Here they are. Oh, yellow, 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 yellow. that's the only one that's going to be anywhere near a match. No. There it is. I don't think you're going to get a closer match than that between paint and paper. I think that's a closer.
We're living in a more colour-coded world, highly dependent on screens. Screens made up of tiny dots of the 3 primary colours. Discrimination and competent colour mixing are key to the way we navigate in this new world. If Lin's colour vision does offer an advantage, could it be that in Lin we're seeing evolution in action?
Clearly, if there was an evolutionary benefit in changing the 2 traditional Al and M pigments to something else, either shifting them in the spectrum or indeed adding a 4th channel, you could argue that we're looking at an evolution of this system, and that in the future we may have evolved into a slightly different trichromacy or tetrachromacy. We may be looking at evolution in the process.
Yeah, that's good.
As culture, art, colour take over as forces for evolution, perhaps we'll see more people like Lin who'll have not 3 but 4 dimensions of colour vision.
They won't come out.
It's certainly plausible that that sort of evolution is ongoing. In fact, it would be the height of hubris to assume that evolution has simply stopped at us as we are now today. There are plenty of improvements to be made.